Imagine trying to learn how a car works to fix any issues that might appear without ever opening the hood, just guessing what's inside and how it works based on how it appears. That's what studying human diseases would be like without the use of model organisms. Since direct human experimentation is generally disapproved before board approval, scientists can't study human health without using model organisms. But how do these different species help researchers evaluate human disease and development? Model organisms are species that scientists study to uncover biological processes applicable to humans. Some examples of model organisms include mice, zebrafish, and fruit flies. But why are these organisms preferred over others? They're preferred because they're easier to maintain, reproduce quickly, and share a surprising amount of genetic similarity with us while being cost-effective. But most importantly, they serve as ethical alternatives to direct human experimentation, allowing scientists to test hypotheses as well as treatment plans safely before human trials. Ethics is an especially important consideration as history has shown a need for balance between the benefits research may offer and participant safety. Cancer research relies heavily on model organisms. Since cancer is a disease of uncontrolled cell growth, studying how cells divide and repair helps us develop better treatments. Think of treatment development like testing a new recipe. You wouldn't serve a dish to others without tasting it yourself first. In the same way, scientists use model organisms to refine treatments before introducing them to humans. We will now share an interview conducted with Dr. Zeynep Öztürk from the University of Oxford to discuss more practical applications of model organisms and introduce a unique mutated organism the lab she's a part of helped develop.
I am Zeynep. I am a postdoctoral scientist at University of Oxford in the Department of Medicine. I did my PhD in Department of Genetics at University of Cambridge in neuroscience, neurogenetics. And now I am working on cancer genetics and cancer mechanism. Our lab primarily focuses on melanoma, skin cancer, and melanocyte biology. For my project, I started working with melanocyte biology primarily. So now I am leading a little bit towards melanoma and neuron interaction. I primarily use adult zebrafish. I culture stem cells or human cells or immortal cell lines in my project. And I also work sometimes with zebrafish embryo. Zebrafish is an amazing model. I think one of the cool things that I kind of fall in love when I saw this model was the transparency of the embryo. So up until quite, when from the embryonic stage, single cell, until the fish gets grown, pretty much good weeks, you can actually see through the animal, transparent, which is amazing. I remember seeing the first video of blood cells going across the zebrafish embryo. It was fascinating, to see that, which is something that you can't really experience in other research models. How we see in the fish. And in our lab, we try to understand melanoma, skin cancer, but also we focus on melanocyte biology, because melanocytes are the skin cells that give rise to melanoma if things go wrong, if they are mutated. And they produce a pigment called melanin. Primarily, we know the function of melanocyte and the melanin as the UV protector in our skin. But research shows that there are melanocytes in different parts of body, not only in the skin, but in internal organs, internal areas, which the sunlight doesn't penetrate. So then the question comes, what are the other functions of melanocyte then? And this is what I actually primarily focus on. And now I am also asking question that what is the interaction of melanocyte with the neurons in the skin, and how this actually related to skin cancer and the development of the cancer. Our lab, my PI, who is Professor Richard White, he has developed this amazing model, which we call Casper zebrafish. And then when two genes mutated in the wild type background, we can see that the stripes are disappeared and all the other pigment cells as well, so including melanocyte, ionophores, and xenophores. So these are the pigment cells of the zebrafish, and when we have these double mutant, we start seeing the zebrafish as a transparent, which gives a lot of advantage on imaging. So the adult fish looking like this, wild type fish, it's not easy to, do imaging or scan through the entire animal once everything intact. But when you look at the Casper fish, you can now see through. You can see this. This is a female fish, and you can see this the egg located here in the animal, and the heart is down here. Once they developed the Casper fish, they transplanted cancer cells into the fish. You can even follow the growth of tumor, which is this melanoma, the black point here, which is actually cancer cells growing, tumor is growing. So melanoma, because it stems from the melanocyte, so it's a pigmented tumor. This is why it looks black. And you can now, in the Casper fish, you can follow and understand more metastatic features of melanoma, and the Casper fish is being a really good model for this kind of studies.